My name is Roland Baron. I'm the co-chair of the Federation. And it's a pleasure for me to welcome everybody to this exciting workshop. Okay, so it's my pleasure to introduce briefly our speaker today, Professor Jennifer Westendorf. She's a professor of orthopedic and regenerative medicine in research. She's also the chair of the Department of Biochemistry and Molecular Biology at the Mayo Clinic in Rochester, Minnesota, in the US. She's been very involved in many of our societies and in the IFMRS, a big data group. And it's really a pleasure and a privilege to have her give us this workshop today. So Jennifer, your turn.
Great, thank you, Roland. It's a pleasure to be here today and to give this lecture and to tell you a little bit about what we've been working on and thinking about in the Big Data Working Group as part of the IFMRS. It's great to see so many familiar faces and names on the screen. I know many of you, not all of you, but I only wish we could be in person in Belgium for the Fleisch workshop, and hopefully soon we'll be able to reconvene that workshop in person. So today I'm going to talk about multi-omics and musculoskeletal research and ways to access and learn from big data. So I'm going to briefly start with an introduction to omics. For some of you, this will be pretty basic, but we'll bridge into single cell omic approaches, and then we'll have a breakout session for you to practice exploring a single cell portal. And then we'll talk about the we'll talk about the musculoskeletal knowledge portal, which is something the IFMRS is developing. And we'll allow you to play with that a little bit, and then we'll have a discussion at the end. So a little bit about me. I'm a molecular and cellular biologist. I got my doctorate here at Mayo. I haven't been here my whole career, but I've been here for quite a while and been running a lab here for the last 14 years. I'm a native of Minnesota, and so that's the state I'm in right now. You can see on the upper right where the TAC is where Minnesota is in the US map. And I enjoy hiking, sporting activities, and being around my 3 nieces. My scientific interests and goals are to advance knowledge of clinically relevant skeletal problems like osteoporosis and osteoarthritis and skeletal tumors. And we do this by studying molecules and pathways that could be therapeutic targets for bone and joint diseases, and by understanding the molecular basis for bone and joint development, degeneration, and regeneration. So that's what I'm interested in. What I'm not, though, is a geneticist or a computational biologist or bioinformatician. That's these skills are very important for big data. So one of the take-home messages today is to, if you don't have these skills, to partner with somebody who has them. And team science is important for advancement of these initiatives into the future. My personal interest in multi-omics that we're going to talk about today really stems from wanting to know more about how the genes or proteins that I'm studying or potentially will study in the future are involved in health and disease. And I'm on the, my disclosure is that I am on the IFMRS board of directors. So an ohm, the suffix ohm, is used in the field of molecular biology to refer to the totality of something. So all of the genes, for example, is a genome. All of the proteins are the proteome. And then omics is a collective characterization and analysis of the pools of these biological molecules that contribute to the structure or function of a tissue, a cell, an organism at any particular time. So it could be very dynamic during the course of the life of a cell or of an organism. And there are many ohms. We talked about the genome. The transcriptome looks at actively transcribed RNA. It could be messenger RNA. It could be long coding RNAs or microRNAs. The epigenome looks beyond the gene to chromatin and DNA modifications. So histone modifications, which are proteins, as well as modifications of DNA. Proteome is for protein. Metabolome is metabolites. That's an emerging field. There's the microbiome, so the collection of bacteria that are in an organism, like our guts. The lipidome, which measures cellular lipids, and the glycome is emerging, which are the sugars and carbohydrates that are linked to proteins and lipids. So those are OMs. So multi-omics is the study of looking at many of those OMs at the same time. So I kind of like the definition on the bottom point here, which is the use of multi-omic technologies to study life in a concerted way. This is a pretty new term in biology. I did a search for PubMed over the weekend, and you can see that there were only 2 references in PubMed for multi-omics in 2006. And last year there were over 1,000, and there have been over 300 already this year. Let's see. I also looked at Google Scholar, and just in the last 2 years, since 2000, multi-omics appears in citations and patents over 13,000 times. So it's definitely an emerging field, and lots of interest in it. And it really all stemmed from the Human Genome Project. Some of you remember this. The Human Genome Project started in 1990. It was the first drafts of the human genome were completed in 1999, 2000, and to great fanfare. We all thought everybody would have their genome sequenced and we'd be curing diseases within a decade. Science really recently just had a cover looking at the genome 20 years later. So it's hard to believe for some of us, I'm sure it certainly is for me, that it's been 20 years since this project started, or 30 years since the project started, and 20 years since it was completed. And you know, it was very controversial at the time because it cost, I think, over $3.8 billion US dollars, but it's generated over $250 billion in revenue, according to a 2015 report. So some takeaways that we know about it is that it certainly promoted a lot of international collaboration and generated large amounts of data, which all of that data drove the development of mathematical and computational tools that allow for the assembly of the genome and annotation of data. And this is very important for the multi-omics that we use today. And so it drove the convergence of 2 fields that really were not working together before. And that being molecular biology and genetics with computational science. And that convergence was very crucial. As I said, there was a lot of hope and speculation that within 10 years of, so in 2010, that we would know the causes of all diseases and we'd be far along in developing cures for the diseases. That hasn't come to fruition in every sense, or at least not to the extent I think that the hope and the hype was promising. But if we look back at the last year, it certainly allowed for the sequencing of the SARS-CoV-2 virus within a matter of days, which is really remarkable to where we were a couple decades ago. It's increased our understanding of history, ancestry, as well as the science of forensics. And it's ushered in this era that we're going to talk about today of other omics and big data projects. We also see new undergraduate and graduate programs in quantitative sciences and systems biology that are being developed in many universities and colleges around the world. It's established the environment for open access and data sharing. And for some of you, it may just seem normal, but this is pretty new revelation for science. So we have accomplished a lot, and I think we should celebrate that, but there are still challenges remaining to meet the promise of the Human Genome Project, including to integrate the data and increasing the accessibility to all that data so we can really understand and cure diseases. And this applies especially to multi-genetic diseases where there's not just a single cause, a single gene mutation, for example, that causes the disease, and to diseases that are not caused by gene mutations or gene mutations alone. So perhaps the regulation of a gene and studying the epigenome or altering the epigenome can cure some diseases. Many of you are probably familiar with that there's many types of data sets and technologies available. We're not going to talk about all of these today, but we will talk about DNA sequencing and GWAS variants. We're going to talk about single-cell RNA-seq. We're also going to talk briefly about ChIP-seq and Ataxic just to provide some examples of how these technologies are moving forward. The spatial transcriptomics, which is located near the bottom here, is really an emerging area that I think we will be thinking more about in the future. So really the basis for all of this sequencing work that we're going to, and big data that we're going to talk about is next generation sequencing, or NGS. And the basic principle here and the revolution here, as opposed to the old-fashioned Sanger sequencing, is that genomes, in this case, or any kind of DNA or cDNA plasmid could be ligated to an adapter. And this adapter would then be hybridized to this bridge platform and amplified. And then using fluorescently labeled nucleotides, you would get DNA sequencing over multiple cycles. And then these reads, although short, you could assemble them into a sequence by looking at the overlapping areas. So these contigs with overlapping sequences would verify that this sequence is correct. And this is how the whole genome was assembled. So you see the assembled sequences here. This is where I don't know a lot about these different types of data files, but this is kind of the data processing sequence. So the raw data comes off the sequencer into a file. It's transformed into a FASTQ file, which lists the sequences. You can generate BAM and SAM files. BAM files are often used to upload into some certain programs that help you analyze data. And then you continue this bioinformatics pathway until we get into a form that I can recognize, and probably many of you recognize, in terms of sequences and expression patterns. So just thinking about DNA sequencing, this is important for GWAS studies. So, one of the hopes was to understand the genome is that we could link the genetic data from different individuals with a particular phenotype. So for example, height. We could say that we know that height is genetically, is a genetic trait. So there are genetic influences on height. So by sequencing different individuals, you could ask if a gene or a region of a gene or a region of the genome was associated with height. And that's been done in a number of studies. And we know there are many genes that contribute to height. But if we look at a single variant as an example, association test with this candidate, which is called RS123, you can see that if an individual has 2 copies of G at guanine at this location, they have a mean height of 155 centimeters. However, if one of their alleles has an A, they're maybe a little bit taller. And if they have 2 A alleles, they're about 5 centimeters taller on average. So in this example, RS293 may be causal, but it might not be. And it might actually be a proxy, though, for a variant that's nearby, in this case, RS456, which maybe isn't sequenceable because it's in a GC-rich region, for example, but it's close to RS123, which is perhaps in the same gene or the same region of the genome. So GWAS uses these evenly spaced proxies for association tests across the genome, and you plot the log fold p-value by genetic region. So this is called the Manhattan plot. You can see the chromosome position here on the bottom. So from chromosome 1 to chromosome 22, and then the X and Y chromosomes are usually on these plots as well. And you can, every dot here is a variant on this particular chromosome, is chromosome 6. So this variant is above this significance line, is associated with height in this case. So GWAS does not mean, because you get a GWAS hit near a gene or within a gene, does not mean it's causing the phenotype that you are looking at, but it does lead to further analyses of regions that can cause the trait and can lead to fine mapping, functional work, and meta-analyses. And typically these studies are done on thousands and thousands of patients, or of individuals, not necessarily patients, but on human subjects or any type of species to identify variants associated with a particular trait. So some of the ways you can functionally validate the importance of a variant include assays like CHIP sequencing, which CHIP stands for chromatin immune precipitation. In this case, you're looking for DNA binding proteins that are binding to a particular region of the genome. So you would that could be histones or modified histones or transcription factors. And so the way this is done is there's a cross-linking step where the proteins are linked, chemically linked to the DNA. You fragment the DNA with the proteins attached, if they are attached. You can do that by sonication or with enzymatic methods. And then use an antibody and immune precipitation method to pull down the proteins. You purify the DNA. So you unlink the protein from the DNA, you purify it. You add this adapter that we talked about earlier. And the adapter contains primer sequences that can be used for next generation sequencing. And then you are assembling the contigs again to the reference genome in this case. So we now know what the genome is. And so these reads can be mapped onto that genome. And then you can see where there are more reads and more hits in your library, you get peaks called. Ataxic is somewhat similar to, or is complementary to ChIP-seq. It's not exactly similar, but it's complementary. But it's looking for open regions of chromatin. So it stands for assay for transposase accessible chromatin. So the key to this is this hyperactive TN5 transposase that has two adapters linked to it. So if there's a open region of chromatin, this TN5 transposase will bind to it and cut those open regions. And these adapters have, again, primer sequences that are known that we can use to amplify these products, purify them, and then again, do the next generation sequencing. And again, where the more open the chromatin is in a cell or a population of cells, you will get peaks. So this can be done, again, on a population of cells called bulk Ataxic, or it can be done at a single cell level as well. Just want to point your attention to a recent article by Terry Cappellini, who did this. Here's the article that was published in Cell last year. He was able to use Ataxic to compare regions of open chromatin in knee articular cartilage, knee chondrocytes, and develop an evolutionary approach to understanding which genes are really important for the development of a knee joint, the development of articular cartilage, and also osteoarthritis risk here. And so he used mouse and human samples and he called it comparative epigenomics, to compare regions that are important throughout evolution. So take a look at that if you have a chance. Moving on to single cell technologies, it's as simple as what you would imagine it would be. So you could take a tissue, in this case, a bone or cartilage or ligaments from a joint. You can isolate individual cells from those tissues. And then you can use the technology of your choice to look at at molecules. So if you're interested in mRNA, you can isolate that and you do single cell RNA-seq. If you're interested in chromatin, you can perform Ataxic. You can even look at proteins with a technology that we're not going to talk about today called CyTOF, which allows you to use a series with up to 30 to 50 antibodies or other probes to look at different cellular functions within on the cell surface or within the cell to really look at protein expression. And the key to this is the way it's represented, these single cell results is this T-SNE plot. T-SNE stands for T-distributed stochastic neighbor embedding plot. It's a statistical method for visualizing high dimensional data. And it does it by giving each data point a location in a 2 to 3 dimensional map. And each data point is located next to other data points that are similar to it. So it's not linear. And it's a way though to condense lots of data into a readable format. And a lot of it's based on probability estimates that, for example, all the cells here in blue are more like themselves than the cells in purple are, but all the cells of purple are more like themselves than they are compared to these other groups here. And some of that modeling is done based on factors that are given to the program and given to the bioinformaticians. For example, if you know what you select osteoblast genes will be fed into the program. Known chondrocytic genes can be fed into the program to help the computer decide what cells are actually being looked at here. So the advantage of these technologies that allows for the discovery of rare cell types or intermediate cell types in a differentiation or maturation process, or even abnormal cell types if you're in a disease state. They may also the data can also be used to reveal relationships between genes and to track trajectories of cell lineages during development. So for this, I'd like to put your attention to an article by Chia Lung Wu when he was a postdoc with Farshkilak. Chia Lung is now an independent investigator at the University of Rochester. This article was just published this year in Nature Communications. And here in a review article, he really describes all of the, at least some of the ways that you can go use a single cell technology, RNA-seq technology. So for example, for cell type identification, you can put these on a trajectory where here the orange cells, for example, are a progenitor cell, and the purple cells are a little bit more mature, and the blue cells start to bifurcate into these turquoise and green cells. So this is all plotted in a pseudotime plot. You can also look at based on receptor and ligand interactions or receptor and ligand expression. For example, if there's cell signaling between different cell types, which I think is really interesting. And then you can look at gene regulatory networks as well. So I encourage you to look at this paper if you're interested in seeing a really high-powered recent article describing this technology. So you may be thinking that technologies and techniques are great, but really, what I really want to do, and you told me this in the polls, is how do I access the data? And the answer, one of the answers is portals. So one of the portals we want to talk about, and this is something new that I just learned about a few weeks ago, but I think it's really interesting. So I wanted to bring it to your attention as well. This is called the Single Cell Portal. It was developed by the Broad Institute, and it's really simple to use. And what we want to do in the IFMRS is encourage more musculoskeletal researchers to contribute their data to this portal. So if you want to log on, please, it works best on Chrome, but I'll just show you briefly here what I did, and we're going to take a little break and allow you to explore it. But just show you what I did and how I think, you know, if you get lost, don't know what to do, you can perhaps try to repeat what I'm showing you here. So simple, very simple. This is the home page. You type in what you want to study, so you can search by studies, or you can search by genes. So I'm searching here by studies, and my interest is bone marrow microenvironment. I get a number of results, 14 total studies come up. They're on 3 different pages. I selected this first one, which is a paper that was published in eLife by Ling Chin out of the University of Pennsylvania, who's on the IFMRS board. And so she submitted this study where she used single cell RNA-seq to identify unique adipose lineage cell population that regulates the bone marrow environment. And so her data are in 7,585 cells. And this is the T-SNE plot from the results. So these are called clusters here. You can see up here, you can download the data if you want. The clusters are named over here on the right side from, I think these are, forget out what all these are, mesenchymal progenitors to osteoblasts, osteocytes. The osteocytes are 5, so it's this orange color down here. You have these unique cells that she identified, the MALP cells in pink, which are here. Chondrocytes are in gray and green up here. And then osteoblasts are purple, I guess, here. So within this, you can search for genes. So I typed in RUNX2 just as an example for you, and you can see that RUNX2 is in just about all of these populations, these 9 clusters that were identified, not so much in the first cluster or the second cluster, but certainly more prevalent in these middle clusters here, osteoblasts, osteocytes. I think these are long-term progenitors and these MALPs. And some in chondrocytes already, which we know, but not as much perhaps in some of the other populations. So that's called the distribution plot. You can also go back to this scatter plot and see where RUNX2 is in these different populations. So I took you won't see this on the screen, but I took this little code, this key from the previous slide. But you can see that RUNX2 in all of these orange dots has RUNX2 expression, and the intensity of the orange is correlated with the intensity of RUNX2 expression. So it's, again, fewer RUNX2 positive cells up here. The gray dots are negative for RUNX2, but a lot more intensity of staining in the other populations. But you also see cells in these populations where there's no RUNX2. Here's another gene, SOST. I picked this one because I know sclerostin and SOST was the subject of the webinar on Monday for the H.Fleisch series, and quite different profile here on this distribution plot, where SOST is really only detectable with this assay in the osteocytes. And then you see that on the scatter plot as well. The only orange dots are down here, again, which are the osteocyte population. Another poll here, just to get your impression of what, get a survey of what you are using and what you're familiar with. Okay, I think I'm going to end the poll here. Okay, great. So most people are, half people are familiar with Ensemble or ENCODE. GTEx is something I use quite a bit. The mouse phenotyping project, the IMPC, a few people are familiar with. A few of you have tried the MSK knowledge portal, which I'm going to talk about next. But about a third of you haven't used any of these. So hopefully you're getting some background and some ideas about resources that are available to you. And one of the places you can go is to the IFMRS website. And there's a big data link on the website, or big data page on the IFMRS website, and there's a big data page that lists a lot of these resources that are available, including ENCODE, Roadmap, Epigenomics, and GTEx. So the links are on the IFMRS page. I find that all of them contain useful information on regulatory elements and gene expression across human tissues. But as you pointed out with the single cell portal, the same problem exists, is that there's limited representation of musculoskeletal tissues in these portals. So the IFMRS and our big data group, one of our aims is to fill this gap and to help researchers identify new treatments for complex diseases, including bone mineral density, fracture, osteoarthritis. Those are the ones we were beginning with, but we're certainly open to other conditions as well. So this is the portal that we are working on. So this is a collaboration with the Broad Institute as well. And the website is mskp.org if you're interested. Again, it works best on Chrome for my use, at least. It does not work very well on Safari. So this is a genomic data mining platform. The aim is to accelerate discoveries for musculoskeletal traits and diseases. And we'll go through how you can use the database. Very simple search function here at the top. So this portal enables browsing and searching and analysis of human genetic and genomic information. So at this point, it's limited to human tissues. And this is information linked to musculoskeletal traits and diseases. And the Broad is very good at doing this while protecting the integrity and confidentiality of the underlying data. So this is the current website is on a new user interface. And this is powered by the Human Genome AMP data and software platform, which the Broad has been developing, especially for their diabetes portal. There's a previous version, but this, if you get to the old version, that's gonna, or you have used the old version, that's gonna be taken off decommissioned at the end of the month. So you'll have to go to this new platform. We currently have 219 data sets and 263 traits represented. On the right side of the homepage here, you'll see some webinars. So the MSKP is part of a larger knowledge portal network that the Broad runs with other disease traits like diabetes that I mentioned, cardiovascular diseases, sleep disorders as well. So we feel really fortunate to have met the right people to get a musculoskeletal portal into this network. We've described, there's a publications listed here that were published last year introducing the portal to the community. And this is, again, a collaboration with the IFMRS and the representatives from these societies with the Broad Institute. But there's opportunities on the website to contact us and provide information and ask questions as well on how we can improve this. And again, this is just the very beginning. We've only launched this about a year ago. This is the first page of the data sets. You can go to the top of the page and click on data, top right of the homepage. These are the data sets. So you see we have a lot of GWAS data from the UK Biobank, for example, the Arcogen study for osteoarthritis. We have estimated bone mineral density and fracture GWAS, most again from a lot of collaborations and consortia are putting these together. There's also data on height and bone size, pediatric musculoskeletal traits, for example. You see how many samples are part of these experiments and these studies. One limitation I would say is most of the ancestry, most of these studies are coming from people of European descent. So one of our goals is to have more diversity into this database as well into the future. On the top, you can do finer searches based on GWAS alone or exome or whole genome sequencing. You can search by phenotype. So here are the other phenotypes that are part of the portal at the moment. So you could look for a gene that you might link in a musculoskeletal phenotype, and it might come up as being very important in lipid metabolism. So it could, by searching using this portal, you could find some unexpected physiological results. And then again, you can search by traits. These are the musculoskeletal traits that are in the portal right now. And then you can also see the data sets and dig into the data sets and find the references as well that are populating the data behind this portal. So there's Ataxic data from human embryonic 59 samples, as well as MSCs. These are HepG2 cells, as well as primary mesenchymal stem cells, and then a number of other data sets. So this is just a snapshot in the top of the list, if you will, for this particular portal. So there are 3 ways to search it. One is by traits and disease phenotypes. So very simple, you click the Explore by Phenotype tab. I typed in estimated bone mineral density. There's also a pull-down menu, so you can search for that and pull down your traits of interest if you don't know exactly what the phenotype is that you're looking for. So what comes up is this Manhattan plot that we talked about earlier for GWAS traits with the chromosomes on the bottom, and then the p-values on top. And so the highest associations for estimated bone mineral density are lying on chromosome 7 here. And then you get this QQ probability plot, which I have to admit, I don't completely understand. But one of the nice things about the portal is that you can, if you have a question about anything, you can click on these links. It's a very interactive tool, and it'll give you definitions about what you're looking at and what's behind the plots. And then a little lower on the page, you see the top single variant associations. So you see these variants on chromosome 7 that probably right up here. You can see that the sequences are C to a T, C or T on the alleles. You get the RS number for the variant. Where it is an intron variant, or this is an intergenic variant. And the closest gene is Wnt16. And then you see the p-values here. You can then zoom in a little bit on just the top common variant with these this particular phenotype. And then you see this list of matching genes. So the CPED1 gene, which I don't know much about, but if I wanted to, I could click on the CPED1, which is in blue here, and it'll take me to a page that'll tell me more about this particular gene. But there are 57 variants near this gene that was identified in these GWAS studies with a pretty high p-value here. And so you can go down the list with Wnt16 is third on this list with only 17 variants in this region. Anyway, you can go down the list and see what genes pop up with estimated bone mineral density. Okay, if you know a specific variant, you can also search this way. So you go to the other tab, type in your variant, and this is the results. So it tells me it's on chromosome 6. The A and G sequences are the normal and the variant that appears. You can explore the region a little bit more by clicking this. Tells me that the closest gene is RSPO3. And this is predicted by the variant effect predictor algorithm. And so you can go look at that, sorry. And then below, it tells you what the consequences that are predicted from the variant. So it's in a protein coding region. I find that interesting. So it potentially has a modifier effect, is if that sequence is different in the gene, it's potential modifying effect on the activity or maybe expression of that protein. Further down the page, you'll get to these what are called PWAS plots or phenome-wide association plots. And this is a locus zoom plot. And here you can see all the musculoskeletal conditions or musculoskeletal traits are in light green here. And the one most linked to this variant is estimated bone mineral density, very high p-value. Bone fracture is a little bit lower, but still moving up a little bit. And then if you look at all of these other traits that are in the knowledge portal or the knowledge network, you see there's one here on lipids, and these are screenshots. If I was on the website, I could scroll over these dots and see exactly which one everyone is. But over here, you can see that there's also an association between waist and hip ratio, adjusted BMI, and just the waist hip ratio alone. And then more of that is listed, those actual p-values for those traits are listed below. So lots of information here, maybe some unexpected links that you would find with your favorite variant or perhaps your favorite gene as well. The other thing you can do is there's a forest plot. I actually like these better. So here you can see that this variant is positively associated with estimated bone mineral density, and these other traits here, and negatively associated with bone fracture. And then the associations with these other phenotypes is weaker. And you'll see up here, there's a code for what the p-values are, or how these colors of these squares associate with different p-values. We mentioned that this was perhaps in a protein coding region. So one of the things with this variant is that this program will pick out transcription factor binding motifs that could be altered, not necessarily, but could be altered by this variant. And this prediction says maybe there's a GATA binding site there, or maybe there's a PAX4 binding site there. So if I saw this, I don't really know what these numbers mean, but I would go back to the exact sequence where that variant is, confirm that these binding sites are potentially there, and then maybe think about following that up with a ChIP-seq experiment or a ChIP-PCR experiment. There are also a number of predictions, and one limitation right now is a lot of the data behind these chromatin and epigenomic associations are from the Diabetes Epigenomic Atlas. And so one of our goals is to populate this with real bone or cartilage or musculoskeletal data. So, but you can see there's some polycomb perhaps, which like the EZH2 proteins could be regulating this variant and the expression of this variant. And again, I'm not exactly sure what all of these things are, but you can, again, dig deeper into this and scroll over some of these things and find the definitions. There are also these pull-down menus that I'll talk about a little bit more later, but you can, on the fly, you can change what you want to look for and what you want to search for. So, well, here's one example. So you can go to annotations. And if you just want to look at DNA sensitive regions, you could really hone in on that. Or if you want to look at active enhancers, you can hone in on that. Chrome HFM, I forget what that is, but you can search for that and figure that out as well. You can also search by tissues, and that is what I did here. So I know there's myoblast data in there, so I searched for muscle. So skeletal muscle myoblasts is what I selected. You can see that right here is the tissue I selected, and with that, the annotation is that perhaps the polycomb complexes are binding to the variant and repressing it. So again, that's something that I would follow up on in the future. And the other thing you can do here is you can see what filter you selected. So if you no longer want that filter, you can remove it and you'll go back to the larger data set. So just more data under the variants. You can see the variant associations in that region that you selected. So this is the particular region of chromosome 6 that's being studied. So this is a summary of the bottom line meta-analyzed associations across the region. So really it means there's lots of variance associated with waist hip ratio and adjusted BMI in this region. You can see total bone mineral density by comparison isn't that high, but it's still perhaps significant here. One of the things which I think has got a little bit out of control, but something we can work on, is all of these boxes down here are the different phenotypes. So the ones in yellow have an estimated p-value of 5 to the -8th. The ones in gray have a lower p-value. So, but if you're interested in osteoporosis, which with this cutoff is not significant, you could certainly click on that and look more at the association of this variant within the osteoporosis data. If these boxes are green, that would be a really high association as well. So green is best, yellow is kind of in the middle, and gray is no association with this cutoff. And then there's this genomic region minor, which I find interesting as well. This is experimental, but allows you to explore variant associations with epigenomic annotations and credible sets across the region. So you can, here's the variant that's highly associated that we search for. So you can see there's a high p-value here. Here's the gene RSPO3. You can also, you can see all the other variants then that are present in the RSPO3 gene. I'll come back to that in a second as we look for particular genes or genomic regions. So again, you can type in your favorite gene in the search bar on the homepage. So I typed in SOST, what you see here is there's a number of genes near SOST that I'll show you in a minute on the chromosomes. But surprisingly, up here on the right side is the top phenotype associated with the SOST gene, which is triglycerides. So I think that's interesting. I don't know much about SOST association with triglycerides. That's been proven experimentally. But estimated bone mineral density is, so there are variants in SOST associated with BMD and with bone fracture. Height, as well as total bone mineral density and femoral neck bone mineral density. So again, down here in these yellow boxes, you see these bone mineral density osteoporosis at a very high level. But you also see associations with knee osteoarthritis, for example, excessive daytime sleepiness. I don't know what that means, but that's interesting. So lots of things that are unexpected, I would say, and things we would really want to explore. So this triangle indicates the top variant that is probably that associated with the SOS gene, but you can see that it's really not that close to the SOS gene and certainly not within the SOS gene. Here's another variant near the SOS gene, but it's also near DUSP3. And then here's another variant in this region. Some things you can do, which I found as I was preparing this, is you can have display options, which will not just show you the dots here and these triangles or diamonds, but it will also tell you exactly what those are. You can also click on these legends here, and that'll pop up, what the p-value is and the significance of these legends. So this is the reference variant that is in the triangle, so that's the highest associated variant. You can take these away. You see right here as well as if you once you set this up and you find the data you want, you can download a PNG of the figure you created for publications. Let's see. Again, very interactive website. You can scroll to the dot and learn more about the variant that's listed there. And then you can even go down to this GWAS catalog, for example, and that'll take you to the GWAS catalog database, which says that this variant is near the DUSP and SOS genes. So you can learn more about the variant there as well. Again, going back to these interactive platform or these interactive pull-down menus, they can change quickly. Again, you can pick a credible data set here that you want to search. You can add an annotation. So these are epigenomic data, but as indicated up here in this little summary, is that these are from the diabetes epigenome atlas. So not from bone tissues necessarily. So perhaps better than nothing at this point, but perhaps not ideal either. But what I did is I searched for a couple things that I'm interested in this annotation. So for example, I wanted to know, are there NF-kappa B binding sites? Are there binding sites for rungs 1, 2, or 3 based on ChIP-seq data or FOS in these regions? And so every place you see a little bar here, there's ChIP-seq data from the diabetes portal saying there's a binding site potential binding site for RUNX1 here. So for example, if we look at the SOS gene, you can see there's perhaps a binding site for FOS and the RUNX factors here, but not NF-kappa B in the SOS gene. If we look at lining it up with this variant, however, here in the middle, none of these factors really align with that. So at this point, this tool, you'll have to use those other tools to try to count on a prediction if there's a DNA binding site, for example, for a transcription factor right here. So you can do lots of things. You can remove these tracks. These are all tracks. So if you don't like the track, you don't want it in your figure anymore, you can remove it. You can move it up and down as well. So this is a pretty flexible user interface and a lot of functionality that I have to admit that I don't know everything about, but it's fun to play with. So this is one of the things we're working on. Again, this is just the start. We have big dreams, and we're setting goals in the big data working group to make this a little bit more user friendly, but also more inclusive of lots of data. So our short-term goals are to add more phenotypes. So we're thinking of more musculoskeletal disease phenotypes, more diversity in the data sets. And we'd also like to add more omic data, specifically for musculoskeletal tissues and cells, and possibly from non-human tissues and cells as well, like rodents. We could even think about data from knockout mice, for example, or knockout fish, or rodents or humans or fish with particular mutations are treated with your favorite cytokine or drug. So we've got this roadmap figured out. Again, we have some disease and traits on the left here: osteoporosis and fracture, osteoarthritis and muscle strength already in the database. It's all human data at this moment, but we'd like to include other organisms. Some myoblast data here, but not a lot on other cell types, and mostly GWAS data. And then the epigenomic data is not right now from musculoskeletal cells. So the long-term goals are to identify and prioritize causal genes and pathways as potential drug targets for risk stratification, disease stratification, or identification. So this is an image provided by the Gemstone project. It's a coordinated roadmap for the functional assessments of functional big data integration for translation to the clinic. So again, starting with existing GWAS data and new studies, also sequencing data from human who have human knockouts where a particular gene may not be functional in a person with a particular trait or condition, mega GWAS data, using bioinformatics to discover these loci, and then to validate those traits and the cause of those traits in animal models or cells and tissues with knockout approaches, transcriptomics, and epigenomics. And then ultimately to identify drug targets, and then, or perhaps stratify subjects for risk of a particular condition or disease. Maybe it even redefines diseases more finely, and we can get different stratification of disease and responses to drugs. This is part of the clinical translational goals And of course, thinking about novel phenotypes as well in terms of imaging and function and novel analytical methods. Certainly artificial intelligence and machine learning is going to be playing a bigger role in this in the future. So again, if you want to contact us or have questions or would like to incorporate your data into this portal, you can contact us on the website. And just want to thank you for your attention, your questions, and participating in the chat. This is a figure we had in the JBMR commentary associated with the paper last summer. So we're happy to take questions, right, Roland?
Yes, yes, yes. Okay, Jennifer, thank you so much for a great overview. I have two things before we take questions. The first is to say that the Federation has been very, very involved and supportive of this whole activity. And we put our money where our mouth is because we have really invested a significant amount of money in putting this portal together. And we are so excited about all the possibilities it offers. And of course, it's thanks to people like Jennifer who spent a lot of time on the big data group, which is really a very good group. My second question is technical, actually, and that is, I see that there is on the broad, of course, for instance, the single cell portal, right? So, and I know, and we've discussed that before, that we do want, and you showed it, to include, you know, all these new technologies, Ataxic, RNA-seq, and so on, and single cell on our MSK portal itself. So how is that going to be done? Is it going to be duplicate? Is it going to be a link?
Yeah, well, we're working, we've mentioned, you know, showing transcriptomic and expression data on the portal. And right now, the Bro doesn't have a way to do that on the knowledge portal.
Okay.
So they're working on it. We've asked them to work on it. They're trying to figure that out, and I'm sure they will come up with a solution at some point. So, but when I saw the single cell portal, I thought, well, that's just beautiful, right? It's simple.
Yeah.
It's I don't know how you can improve on that. I'm sure there are ways to improve on it. There's probably minor things you can do, but you know, for me, that's so simple. And I think the knowledge portal already has a lot on it, and you have to dig deep to find things. But it's probably going to need multiple tool sets, to be honest. Maybe in a decade, though, you know, we'll have a machine learning program that spits out all the data for us in 5 minutes, right?
Yeah, it's a really fascinating time, really. Okay, we got a question here. My gene of interest does not appear to be important for any MSK phenotypes in the portal. What should I do? Should I stop working on this gene or what?
Yeah, I would say no, definitely not. If you've got you know, some data from a knockout mouse or a functional assay that a gene or a protein is important, continue to work on it, because it may not rise to these high p-values that you know, that are linked and that rise to the top of these lists in the GWAS studies. And as I told you when we're talking about GWAS, is that this is just a variant that's associated with a trait or a phenotype. Doesn't mean causality at all. So, especially for multifactorial, multi-genetic diseases, it might be a series of genes that, you know, individually have low p-values, but collectively are important for causing the disease. So you might still be onto something if you've got good functional data for your gene of interest.
Yeah, let me share with you one of my frustrations. And actually, I see Rick Sumner asked a question about Win16, saying it's highly associated with BMD and fracture. Is there a way to tell if low BMD is associated with fracture for this gene? So my frustration each time I've been looking at these things is how do you go from the variance to a functional assessment?
Yeah. Well, you know, I think there's a couple ways. If it's in a coding region, that's easy, right?
No, sure.
answer, we know what to do, right? We do some cloning and we introduce the mutation and we do our functional assays, right? If it's in an intergenic region, it's much harder. And that's where these 4C capture techniques can play a role, because we know that DNA is not linear in a cell, and that the DNA will loop, for example. So something that seems far away actually might be really close in 3 dimensions within a cell. So it could be regulatory. And so there are these 3C, 4C capture techniques that allow for the identification of the regions that are actually pretty close to each other inside of a cell or inside of a nucleus. I didn't have time to cover those, but that's perhaps a study for, or a topic for another webinar.
Sure, but most people are not going to engage in that. The risk is too high. I mean, it's just a variant. The risk is very high to figure what the functionality of it is.
Yeah, and there was a paper recently by Kurt Hankinson's group and others that were doing this in fracture, I believe. So this is coming. People are starting to look at this, especially trying to understand these GWAS associations and GWAS hits with fracture and osteoarthritis.
Yeah. Yeah, and in the single cell analysis, how reliable do you think these pseudo time things are? Because I understand that you take, okay, what is most mesenchymal stem cell, like, and then what is most differentiated like, and then you can, of course. But what about all the in-between steps?
Yeah, it's a really great question. I think that they can be hypothesis generating, right? So what I've seen people who have published papers on these is they've said, okay, we're gonna take this information and then we're gonna use mice that have different reporters on them, different fluorescent probes, and see if we can really validate that trajectory in mice during development. So I think that's where the strength comes, is this is hypothesis generating and can lead you to experiments that will prove it. This is, a lot of this is coding and programming that I don't understand, I have to admit. And so there could be biases. I think you bring up a good point. In these computational methods, the computers are only as smart as we tell them to be. And they only look for the things we tell them to report on. So I think that this is where, for the young people on the call, is to get training in both, maybe specialize in one area, either biology or computational sciences, but be able to speak the language with a collaborator. So to move this field forward, I think that's really what's going to be needed in the next generation to make giant leaps.
Actually, we have another question here. What if my gene of interest doesn't show up in a single cell RNA-seq plot? So it's the same question as with but for single cell.
Yeah, I think again, it's it depends on the size of that. You have to look at, again, the details of that experiment, how many cells were analyzed. That's typically not very deep sequencing at this point either. So they're really going to just be pulling up the most highly represented genes in that single cell seq. So that may change in the future as the sequencing becomes more efficient and deeper. So I think you really have to look at the details of how that data were generated by going back to the papers and seeing how deeply they sequenced it, if you're concerned about it at all. But I still like to validate things with PCR, qPCR, protein levels. Actually, it's my favorite way to go.
And look, as we are all venturing into these RNA-seq bulk or single cell, there's a question that I hear often from the people, the companies and so on. So do you want depth, or do you want, I mean, can you make some comments about that? I mean, it looks like a trade-off to me, and I'm not sure what we are sacrificing one way or the other.
It's a good question. Usually it comes down to cost, right? But we know that the costs are being reduced, they're becoming lower every year and decade. So
We hope.
we hope. I think that's probably the major factor, right? And people driving these decisions. Is it best to get something out, or is it to go deep? And again, I think that's a personal preference, right? What are you really looking for? What's the nature of the experiment? So ideally, we'd like to have it really deep and really cheap, right?
Ideally, you want to be deep and cheap.
Or deep and inexpensive, I should say. But you know, I think we have to make, you know, these are trade-offs, as you say, that we have to make decisions about.
Okay, anybody else has questions? You know, I could talk with Jen for hours, but I doubt that that's the right solution to the problem.
Anything, I guess the question is, how can we make the portal better? You know, is it, how would you rate it on a scale of 1 to 10? And if it's not a 10, what do we have to do to make it a 10?
Yeah, that's a good question to ask. Yeah. Well, for me, I mean, if we manage to put in there not only the GWAS that we have, but also the bulk RNA-seq and the single cell RNA-seq and whatever proteomics will pop up in the next few years, then you will have a portal where you can establish links between these databases. And I think that it's going to be fascinating. But like you, I think that every department of biology will need true bioinformaticians, because it's not that simple, especially to analyze, I mean, to generate data. Now, okay, you generate data, but you know, from my own experience, if you design an experiment that is, you know, physiologically relevant, then you're going to have several groups, if you do animal studies, for instance, and then you get a huge database for each group. And it's not that simple to get into that and make sense out of it. No,
Yeah.
so it's going to be.
I agree. So yeah, Debs, it is a complex portal. So, but I encourage people to go back to it. If you come up with a new gene or come across a new gene that's of interest to you, check it out in the portal. We're going to continue to improve it, keep coming back to it. So keep playing with it, keep coming back to it. Yeah, and Marilyn, yes, one of the reasons we're doing this is to get the word out about the portal. So thanks everybody.
Yeah, a lot of comments.
Yes, and I encourage you, those of you who are on the call, I see lots of people who are part of the IFMRS societies. So please spread the word to your colleagues within your societies.
Yeah, and it shows the strength when all the societies start to work together, and that's what the federation is all about. So I'm really excited about all that. Okay, so thank you so much, Jen, for a great presentation. I mean, I can't believe that we can do almost hands-on workshops with Zoom. And I always wonder, how would the pandemic have been if we didn't have tools like that? I mean, it would be scary to death. I mean, it's unbelievable.
Right, this last year would have been much harder without this technology, for sure.
Yeah.